Hi, welcome back to the second Q&A session here. I'm pleased to be joined by Megan O'Rourke and Drs. Akot, Hasted, Proel, Breitschwart, and Embers. Welcome, everyone. Thanks for taking the time. So as we said before, if you have questions, we're having a little problem, technical difficulties with the Q&A section. And so if you want to send in a question, you can send those to LymeMIND at steveandalex.org. So that's LymeMInd at Steve, S-T-E-V-E, and A-N-D, Alex, A-L-E-X.org. All right, so why don't we jump right in. So one of the first questions, and I think this is more to the first panelists, was around mast cell activation. Could someone talk about mast cell activation? Is it downstream of infection or a cause that makes infections worse? And a sort of a follow-up was, would that have a potential to be studied at the clinical trials network that we talked about before?
Sure. I'd be happy to talk a little bit about MCAS. MCAS, or mast cell activation syndrome, is a condition in which patients find that their mast cells are degranulating, and mast cells are innate immune cells that are releasing a lot of mediators. And if there's overexpression of these mediators, it can cause a range of symptoms, from those that are more classic, like allergic type symptoms, to now symptoms that are common in chronic or post-treatment Lyme or even ME/CFS cases. Now, one thing that's important to understand, though, is that organism and mast cell, their activity is intricately connected. So mast cells will respond to organism in any, so if there's organism in tissue, for example, mast cells will come in and respond to that organism, right? It's part of what they do. And interestingly, if organisms form into biofilm, so sometimes let's say there's bacteria in a tissue, those bacteria will team up together and they'll form a little community in which multiple of the bacterial species are in this protected matrix together. So there's actually studies that show that biofilm signaling molecules in these communities themselves activate mast cells. So one thing is, if you have an ongoing untreated infection, whether it's a single pathogen or a group of pathogens, such as a biofilm infection, that will be a perpetual stimulus of mast cells that can lead to at least some part of the activation that could contribute to MCAS. So that's one thing I think to take into consideration is, when I hear that someone has MCAS, sometimes I go, do you have an undiagnosed infection that you might need to consider as part of this clinical picture?
The answer regarding the clinical trials network is absolutely. We're open and been seeking input on ideas around all aspects, both pharmacologic and non-pharmacologic, and including aspects like mast cell, as well as infection itself, as Amy was pointing out. So the answer is yes, we are interested.
Great. Another one for you, Dr. Akot. What specific experiments to show actual effects of infection or nerve brain at molecular level rather than just observational?
So we've published observational and molecular evidence of brain involvement in Lyme disease. So the molecular level, the PET imaging studies involve a molecular imaging tool that binds to activated inflamed glial cells, and it's actually, at the molecular level, it's a specific molecule that binds activated glial cells. So that's an example of using imaging on a molecular level. We're also currently funded by the DoD to do an fMRI study that includes cerebrospinal fluid that will also have fluid that can be analyzed at a molecular level. So it's really important both with imaging and getting cerebrospinal fluid, and that's what we're in the process of doing now.
Great. I guess a sort of more general question, maybe this one's a good one for Megan. How do we ask our doctors, general practitioners, to help with treatment of Lyme and sort of this idea of sort of the reservoir bacteria, so someone with maybe a more persistent infection?
Yeah, that's such a great question. I think the challenge here is to create a relationship of trust with your doctor, right? And to find a doctor with whom you can have a relationship of trust and listening, a doctor who listens to you. Beyond that personal connection that you find, pointing the doctor to some of the specialized literature that you've been hearing about here at the conference, that you know about, showing that, the standard 2-week course of doxycycline or 3-week course of doxycycline isn't a one-size-fits-all prescription for all patients, right? So really pointing them to some of the great literature showing the variety of outcomes, or suggesting at least a variety of outcomes for Lyme and tick-borne diseases.
Great. One question here for Dr. Embers. For adults with non-Alzheimer's dementia symptoms, how should their doctor test them for co-infections such as Bartonella? And if there's any, they're saying they're only tested for Lyme so far.
Yeah, I think comprehensive testing is important with any case of Lyme disease, because we know that co-infections are more common than not. For Bartonella in particular, it's important to look for Bartonella in the blood, and Dr. Breitschwert can attest to this, because a lot of people will have antibodies. And so specialized testing for Bartonella in the blood is very important.
Great, and I guess one other question around Bartonella, what are sort of the common symptoms or things that people should be looking out for if they might be suspecting Bartonella?
I think there's kind of two ways to think about Bartonella. The acute infection, which is classic, is cat scratch disease, where a cat scratches an individual. The organism is actually transmitted through flea feces that are inoculated at the site, and shortly thereafter, fever, lymphadenopathy develop. And then I think most people clear that infection at that point in time, independent of antibiotics or with antibiotics. When we start talking about chronic infection, which has certainly been the focus of our research, the symptoms just vary tremendously. And our IRB questionnaire has 2 columns and about 40 things that people can check in regard to symptoms. And some individuals that will test positive after being sick for several years will only report headaches and insomnia. Other people will literally check every box in 2 columns. So the symptomatology can vary and can involve any organ system in the body.
Interesting follow-up here too. Does anyone want to talk a little bit about the relationship or potential relationship between Lyme, tick-borne disease, and cancer?
Well,
and sort of the research around that.
What I usually go back to is the World Health Organization currently accepts the fact that nearly 20% of all human cancers are directly caused by infectious agents. The emphasis, however, has been on viruses almost exclusively, with the exception of Helicobacter pylori. So my bias is that intracellular bacteria that persist for a long period of time and behave like viruses may end up playing an important role in cancer causation. But we're just at the beginning of that story.
Yeah, and I would like to add that I think an active area of very recent research is actually the tumor microbiome. In other words, all the communities of interacting bacteria, fungi, viruses, and even bacteriophage, which are viruses that infect bacteria and modulate their activity in tumors. So there have been about at least three large studies that have used new computer-based tools to find a range of organisms. And these tissue types were considered to be, in many cases, sterile, and they're actually full of organisms, sometimes dozens of different bacterial pathogens. And some of those studies have focused on GI tumors because it's actually easier to get that kind of sample. But fusobacteria has been identified in a growing number of bacterial tumors. And there are overlapping ways in which Borrelia, fusobacteria, and other bacterial pathogens can drive cancer. So there is actually a real growing interest in bacterial contributions to cancer. And I think Borrelia and Bartonella can be better studied in that capacity.
Question here for Dr. Hasted. Will there be any studies on the pharmacology of the antimicrobials regarding biofilm and biofilm-like aggregates in tissues for these diseases like Bartonella and Borrelia?
Are you talking about our drugs, or we do know that the one we're working on does recognize the protein target in biofilm, and Dr. Spector's lab had a nice paper where they allude to some of that, I think, in some of the data that came out this year or early or late last year. You might want to check that out. So many of the the organisms secrete many proteins, that form the biofilm, and some of those are druggable, and one of them happens to be one we work on. So I think that could be of diagnostic importance, obviously.
Great. Going back to Dr. Prohl, any vagolytic experiments to block effects? I'm guessing that's
a good question. I think when it comes to vagus nerve signaling, it depends on how you want to modulate signaling of the nerve from a therapeutic perspective. So there are a number of companies that create what are called some vagus nerve stimulators, or VNS for short. And in one model, for example, I actually have one. You can attach it to the inner ear, and then it creates a small pulse of energy that affects the vagus nerve. And it depends. There are researchers who are experimenting using that kind of stimulation under different settings. So for example, because you may want to enhance signaling in vagus nerve during a time when the patient is already doing something else that is related to the disease process. So for example, in PTSD, they're considering using stimulation of the vagus nerve during a session in which the patient works through some of their memories during some of their issues in order to help condition the nerve to better help signaling from the body to the brain be more, sort of modulated correctly during that period. Another example would be in patients with gut issues, where vagus communication between the gut microbiome and the brain is a big area of signaling. And so, for example, you could use a vagal nerve stimulator during periods of peristalsis when you want the gut to be moving more, for example, in order to encourage that. And there are some teams that are beginning to use vagal nerve stimulators in scanners, in other words, in imaging scanners, so that they can challenge the vagus nerve there and actually see via imaging what's happening to the patient's brain and body when that happens. And I think that's going to help us gain information on how vagus is impacting these diseases, and then how we can use vagus nerve stimulating type devices to better intervene.
Great. And the Cohen Clinical Trial Network is actually considering a vagal nerve study at this time as a potential pilot study. So we're actively looking at that.
That's great. One last question for you, Dr. Arcott. Based on what you're seeing, do you have hopes that treatment of persistent infection might help, particularly with brain inflammation you've documented?
And we're very interested in certain antibiotics that have combined antibiotic and anti-inflammatory effects. And some of these antibiotics are antibiotics, but they also calm down inflammation, specifically glial inflammation, which is one of the mechanisms of brain, inflammation and symptomatology. So the answer is yes. We're very interested in combining actually those two important effects, both antimicrobial and anti-inflammatory properties. And there's several actually molecules that do both of those things that are very interesting to us.
Well, great. Well, thank you guys all for being part of the panel today, and also taking your time over the weekend to participate in the Q&A. I will just remind the audience too that all of these will be all these panel sessions and Q&A will be available for replay on LymeMIND.org in the next few days, probably a week. We'll be posting them. So thank you all for taking the time.